All right, I think we can get started first, and then people will show up later, I think, usually. Okay, welcome everyone to our bioinformatics and genomics seminar today. We're very glad to invite Dr. Julien Gagneur from Technical University of Munich, to talk about his recent paper, Detecting of Aberrant Event in RNA-seq Data. And for the next month, we have two topics about COVID-19. So they publish in the Science and Nature Medicine journal. So if you are interested in any of these topics, you are welcome to join us. And for all the previous seminars, we actually recorded the videos in YouTube. So if you miss any previous seminars, you're welcome to come to this page and click the link. You can listen to the talk in YouTube. Dr. Chris Euskastis from BCH will co-host this meeting with me. So I will turn it over to Chris. Thank you, Chris.
All right. Well,
you can introduce our speaker today.
Yep. So it's my pleasure to introduce Julien Gagneur to give this talk. So he currently is the full professor as of last year. Congratulations. Full professor of computational and molecular medicine at Technical University of Munich. He initially got his PhD in applied mathematics at École Centrale in Paris. He then became a computational scientist during that time at Cellzome and Heidelberg, and then a staff scientist in the European Molecular Biology Lab in Heidelberg, before becoming a group leader in Celgene, and then ultimately joining the TMU as an associate professor in 2016, and then has risen through the ranks as now a full professor. His work spans multiple areas of really bioinformatics, from looking at different techniques for really understanding transcriptional regulation, and they pioneered the transient transcriptome, or TT-seq mapping, and then had a recent Nature biotechnology on this KIPI repository for predicting models of genomics. And then his paper that he's going to be talking about now is really exciting to really understand or detect aberrant events in RNA sequencing data. So we could potentially use these for help in understanding with our gene discovery techniques using the RNA sequencing platforms as well. So looking forward to hearing his talk.
Thank you. Thanks for the invitation. And I was pleased to see this list of talks. It's really great, and will be also a resource for the lab. So thanks for putting this up together. I hope you can see now the presentation. Full screen, correct?
Yes, we can see that.
Excellent. Yes, so my talk will be indeed on genetic diagnosis of Mendelian disorders using RNA sequencing. And first, let me, in a very few slides, give you a little bit of an overview of what my lab is doing. So we're interested fundamentally in progressing in our understanding of the genetic basis of gene regulation and its implication in diseases. And indeed, as you mentioned in the introduction, we work with basic biologists to have better genome-wide measurements of different steps of gene expression, from transcription all the way to translation. And then based on these data and also publicly available data, we build models that predict any of these rates or steps of gene expression from sequence, with the goal eventually to apply it to interpreting genetic variation in rare disease, but also now we're embarking a bit on oncology. So just to highlight two things from this work. One model that's became a bit popular is called MN splice. So it's a deep learning model which we could train on massively parallel reporter assay data, and that won a challenge back in 2018. And recently, the CAD team benchmarked several models, including MM-Splice and Splice-AI, and both MM-Splice and Splice-AI stood out as the two best models, and they include now this into CAD. So it might be helpful for you guys to interpret variants that are plus minus 100 nucleotides from an exome. So the publication is here. And then indeed, I'm happy that in the introduction you highlighted KP. This project is a collaborative project with Anshul Kundaji and Oliver Stegle so far, but any other labs, I'm happy to join. What we are trying here to do is a standard repository of trained predictive models such as MM-Splice, Splice-AI, or others, in a way that is standard, so it's become easy to apply directly on, for example, on VCF files and annotate VCF files, and so that we have in a way an acceleration of this knowledge transfer from people working more in regulatory genomics all the way to clinical settings. It's also the standard might also be helpful to develop new models. So feel free to have a look at this repository and maybe also join our monthly discussions on KP. But despite all the progresses of this field of regulatory genomics, as of today, at least in the context of Mendelian disorders, we have a kind of plateau, a capping of diagnostic rates when we try to interpret just genomes. So the majority of individuals after genome sequencing with a suspected Mendelian disorder remains undiagnosed. We cannot pinpoint what is the genetic cause of those diseases. So the DNA is not alone, it's not enough by itself. And so what we have embarked on is a long-standing collaboration with Holger Prokisch, a colleague here in Munich, with whom we are developing techniques that are based on functional genomics assays and that complement the DNA information to accelerate the diagnostics in context of rare diseases. So Holger is focusing on a class of disorders that is, in a way, very practical to develop such assays, and these are mitochondrial diseases. And why are they interesting for establishing functional genomics-based diagnostics? It's because of the following. These mitochondrial diseases are defined as disease that affects oxidative phosphorylation, so cellular respiration. They're quite prevalent, affecting 1 in 5,000 births, so it's one of the largest metabolic disorders. And interestingly, the number of known causal genes is pretty large. There's up today over 340 known causal genes. So mitochondrial disease are not only due to the 13 genes encoded in the mtDNA, but they are mostly caused actually by genes in the nucleus. And this list keeps increasing. We still have not a complete list of known causal genes for mitochondria. So that means that when you look at an individual, you probably find maybe a new gene here or variants in one of these 340 non-causal genes. Symptoms can manifest in various tissues. There is some prevalence to tissues that are highly demanding in energy, such as tissues from the CNS or muscle. However, you can find the phenotype also in other tissues, and this is because mitochondria are expressed in nearly every cell type. So one can work actually on skin fibroblasts and assay cellular respiration, even though those patients don't have necessarily any pathological problems in skin. And that also means that complementation and supplementation directly in patient cells can be done quite quickly. So one can validate hypotheses with relatively quickly experimentally. So we embarked a few years ago in asking how transcriptome could complement all exomes back then for the diagnostics of rare disorders. And to do so, we made a pilot study with over about 100 individuals. Half of them were already diagnosed from whole exome sequencing analysis and have not. And from the fibroblast, we perform RNA sequencing. And here the concept has been to focus in the transcriptome in three different patterns. The first pattern is to look for aberrant expression. So the idea is you're looking at a rare disease. So here you're not in DNA, you're not going to look at a rare variant, but you're going to look at something rare in terms of expression. So typically a gene which in one particular individual is at a level of expression far away from the physiological range. We've been focusing mostly on underexpressed genes, but you could, in other contexts, say in developmental disorders, definitely in oncology, also look on the other side, on genes that are overexpressed. So that's one pattern. Another pattern is aberrant splicing. You may have exons that you usually do not see and lead to severe defects on the gene, especially if they lead to frame shift or encode a stop codon in frame. And then the third pattern has been monoallelic expression of a rare, potentially deleterious allele. And this is important in our case because in these metabolic disorders, we often take the assumption of a recessive disorder. And so such heterozygous variants will not be prioritized from DNA analysis, but could become prioritized if one observe that only the deleterious allele is expressed. And as I said, we enjoy the situation that these assumptions or these findings can be quickly validated directly working on patient cells, on skin fibroblasts. We've done that with complementation and supplementation assays. And in this very first study, we increase our diagnostic rate by 10%. And I'm going to tell you today about results that we have with an extended data set of 305 samples and with new methods that lead us now to even an increased diagnostic rate by 15%. So 15% on top of what you get from the whole exome sequencing, that means that you are moving from half of the individuals being diagnosed to 2/3. So the first set of genes that the first pattern that we're going to look at now is aberrant expression. And this is based on a method that we published a bit earlier called Outrider. So there are two particular issues on a statistical point of view that we need to tackle in this detection of outliers. First is that if you use a classical tool for differential expression analysis, such as DSeq or EdgeR, you're using a tool that has been actually developed for a different goal, not for detecting outliers, but rather for assessing differential expression between groups. And those methods have been actually even optimized to deal with small sample size, so we're having a few measurements in each of the two groups. In our application, we have a different situation. We have a cohort of 100 samples. You will collect more in the future. And what you're actually interested in is finding data points that are far away from the global population. It's a different statistical question. The other aspects that we have to control for, and that's pretty general in differential expression analysis also, is to be able to control for potentially unknown common source of variations. What I'm showing here on the right-hand side is a correlation matrix of our samples. And what you see clearly, as you would see in any compendium of transcriptome, you have blocks of samples coming together, samples that are highly correlated with each other or less than others, and even different degrees of structures here. And so if you do not control for this, what you see on the left-hand side is not such a nice separation of your outlier from the cloud. Is actually multiple clouds, and you really need to know to what cloud your data point belongs to. So we need to do both at the same time. And to make very clear here these points of the common source of variation in the context of rare disease, you get to see that controlling for these common source of variations allow you to enrich for rare gene regulatory effects. So what you're controlling for here are, on the one hand, technical biases, that may be known or can be unknown. You also control for common genetic variations, such as sex, ethnicities, and so on, which can have also these effects on gene expression, and common environmental variations. So once you control for that, gene regulatory effects that are very rare will stand out. And that's exactly your goal here, to identify in the transcriptome such rare effects. The technique we've been using is a technique borrowed from signal processing, from machine learning, called denoising autoencoder. And it's nicely explained with images. So I'm using now the application of computer vision to explain you the concept of denoising autoencoder. What you want to do here is to identify also outliers, with the goal rather to remove them from your data. We are interested in finding them, but it's essentially mathematically the same. And the strategy that denoising autoencoders implement is the following. You start from a database of, in this case, images. So it starts from your different instances of your data. And then what you do is you artificially, in silico, corrupt your data. So we add noise to this data. And then we ask a model, which takes as input the corrupted data, to retrieve the original data. So you're predicting what is the original data, taking as input a corrupted data set. And so the delta, the difference between the two, will be the outliers. So this has been well developed in machine learning. It's essentially based on a squared loss, and we had to adapt it for transcriptome. This is what we've done here. Instead of taking least square error, we took a loss function based on the negative binomial distribution. That is a distribution well adapted for read counts from transcriptome. Otherwise, we use the same concept. So that means we're going to do a dimension reduction, a bit pretty much like what a PCA does, to model all these sorts of covariation, and then ask for the contrast between what you can model jointly from what stands out from this joint modeling. So technically, visually here, it would mean that if a gene is at a relatively high expression level in the context of a certain samples, you can detect that. That is something that you would not be able to detect as an outlier if you were looking at expression level of that gene marginally without controlling for these covariations. So we do this, and then we have a test based on the negative binomial distribution. When you do that, we have then p-values, which allows us also to do multiple testing correction, something that if you just apply z-scores out of the box, you may not be able to do nicely. We were also able to check proper calibrations of our p-values by QQ plots. This is an example of a particular gene. And then what our package outputs are plots like the QQ plot, and also plots like this one, where we show the normalized counts on the left-hand side, and all those sorted by expression levels. So an outlier here, we see two instances of under-expression outliers that are really down compared to the rest of the population. We could validate this not only by simulations, but also by looking at enrichment for rare variants. So the idea being that not all rare variants leads to aberrant expression, but you should see an enrichment for rare variants among those outliers. And we found that indeed our outliers are enriched for such rare variants in GTEx. So this is every data point here is one of the 50 GTEx tissues. We find an enrichment that is consistently better than if you do PCA for your correction or PIR for your correction. And this is true for this p-value, but also for other cutoffs. So we like our methods, we can also check for generally for enrichments of variants. So this is done now here on this data set of over 300 samples from individuals suspected to be affected by a mitochondrial disorder. And what you see nicely is that among the underexpression outliers, over 35% have a rare variant detected by whole exome sequencing. And if you break down this by VEP variant annotations, you will see that annotations that associate with nonsense mediated decay, such as stop, frame shift, and probably some of those splice altering variants, constitute most of those variants. So this tells us that we probably have indeed true outliers, and we can also combine data from genetic and from transcriptome to pinpoint a variant that indeed has an effect on gene expression. So here's an example. This is an example from this first publication. We found 2 individuals with an aberrantly expressed level of the gene TMNDC1, which back then was not yet a disease-associated gene. And TMNDC1 is a protein that helps the respiratory chain complex 1 to assemble. At that stage, we didn't find any rare variants. You will see later with more slides on these cases. But we could spot aberrant expression, and we could also complement this expression of TMNDC1 and retrieve respiratory activities of fibroblast cells by overexpressing a wild-type version of TMNDC1. Here's a more recent case that I like to show you because it shows you also the sensitivity of our trialer. We have here a downregulation of a gene, METC2, that is by about 40% down. Well, it turns out that this is an individual which has a de novo, so a heterozygous stop variant that affect one of these alleles. We could check this by looking also into the parents and then validate in collaboration with this group here the function of this gene. So that shows you that also for aberrant expression by only 50% or less, you can get an outlier. Look here, this point. After a proper correction of all the common variations, one is sensitive to those levels. So this can be helpful for dominant cases. Okay, now let me go through aberrant splicing. You will see that the concepts we've been applying are pretty similar. It requires, however, a particular treatment of the data beforehand that is a bit more advanced than what you do in gene expression. Okay. So it is important in the case of splicing to look beyond annotations because you may have a novel splice site created in a particular individual. So we and everyone working in this field are using then methods that directly call splice sites from the RNA-seq data by looking at such so-called split reads that show evidence of splicing of an intron. So by looking at split reads and having accumulation of split reads at a particular position, one can de novo define introns and therefore donor and acceptor sites. So that's the first step of our method called Fraser. And then we decided to look at a metric that has been introduced in the field by Roderick Gigo's lab called the Psi5, or person spliced in of the 5' end. There's also a Psi3 for the 3' end. It is defined as follows. You consider a particular donor site, and then you look at all the reads from this donor site that goes into a particular acceptor site, all the split reads. And you look at a fraction of those among all the split reads that start from this donor site and go to any acceptor site. So this is this fraction here, which is between 0 and 1. And that's Psi5, and you can do the same on Psi3. There is another related metric also defined here in this paper, which is called the splicing efficiency. And the splicing efficiency still consider all those splice sites. And then now look at the non-split reads, so the reads that go from the exon into the intron, or on the acceptor side from the intron to the exon. And here what you do is you look at a number that would be 1 if splicing is complete. So if we only see split reads. So what you're building is the sum of the split reads over the sum of the split reads plus the non-split reads. You can define that again from the 5' end or from the 3' end side. And those metrics are pretty simple to define. They do not require the complete assembly of a locus in terms of all splice sites and all donors and acceptor sites. It doesn't require neither an estimation of splice isoform as you have from some algorithms that are based on that. That means it needs very little data preprocessing and very little mathematical assumptions. And it turns out it's actually a pretty robust way to deal with this data. Okay, another point which was maybe a bit surprising and was not always considered in the early days when people were looking into aberrant splicing is that there are actually a lot of covariation. So even though those metrics are fractions inside one sample, you could say, well, I control for everything that can happen. It is not true. There's still a lot of covariation of those metrics between samples. So here we're selecting the most variable sites, and we're building a correlation matrix. And what you see is that just like for expression levels, there are groups of samples and even some kind of hierarchies of groups. So again, there can be different source of variations that affects splicing. Some one can think of, others we don't know, and non-confounders. We've done this across multiple tissues and GTEx, and sometimes we can relate it to the sequencing site, sometimes we can relate to the degradation index or to the cause of the disease, but it's very hard to find a common topic everywhere. So we let, again, a denoising autoencoder do the job for us, model all this called covariation with the recall of outliers as a criterion to fit the model. And this turned out to work well. So this is now a correlation matrix before and after correction by the denoising autoencoder. So very similar to what we've done for expression outliers, we can do now an enrichment for variants that are predicted to affect splicing, either because they lie on the splicing region or because our model MN splice predicts it. And what you can see is that with Fraser, compared to alternative models that could be more naive or that we've done in the past or that others have done over the last year, we have a better enrichment. We've been a bit disentangling the reasons for that, and it's really this fitting, especially against this alternative method here, it's the fitting of the latent space that seems to be the important feature. And you see it's backed by genetics. This is done really with independent data. Now we can do, again, we can ask what are the variants that we find on those outliers. Here, the enrichment is a bit lower. When we look at whole exome sequencing rare variants, if you have access to larger data sets of whole genome sequencing, you may be a bit more enriched. So we are at about 10%. And these have the types that you would expect. So we find among these 10% variants that are either close to the splice site or in the exon. And you know that many regions in the exons can affect splicing, and also intronic. These results are similar to results that the GTEx group have done with a different tool. And so we are finding essentially the same things here on individuals with a suspected mitochondrial disorder. Here's, we're going to follow up this TMM-DC1 example, which gave us a lot of insights. So, let me show you how what we learned from studying this aberrant splicing. So Fraser identifies a aberrant splicing on 2 individuals. And this is what you see here now is an IGV view of this locus. The y-axis are RNA-seq level. This is a so-called sashimi plot. So what you see, the numbers here are the number of reads supporting a particular intron. And what we find in these 2 individuals, in contrast to these two other samples, there are 100 of those that we could plot here. Those two are just representative, is that we have a new exon which is outside the canonical annotations of this gene. We can do more. We can look into the transcriptome and actually find a variant in the transcriptome, exactly on that exon. And that variant is homozygous in this individual. We also performed whole genome sequencing and found it only in those individuals. Now, the exon, which this novel exon has a stop codon in frame, and so it leads to a truncated protein. And this is something we could validate at the proteomic level and then also complement, as I mentioned before, showing that this was causal for the disease. Now, the surprising thing, or the difficult thing, if you come back to here, is that if I tell you there's a variant deep intronic here between the intron 5 and 6, even if it's homozygous, you will have a hard time to prioritize that. A model like Splice would not capture it if you do not tell that you have to focus here, because it's too far from canonical exons. So how do you do that? Well, we have a bit of insight when we look at the very same location. On the other samples, we observe there's actually weak splicing. So this exon that is not annotated is actually a place where weak splicing occurs. And so the implication for that is that we should actually look at those locations when we prioritize variants. There's something we're following up now. We don't have this completely wrapped, but it's indeed a good idea to annotate variants, not just with respect to canonical variants, but also looking at locations where there's weak splicing occurring. Just a bit of data. We've been following up a bit this idea in the Kramer paper, and we found that for 80% of so-called private exons, so exons that we found in 1 or 2 samples of the 100 samples, for 80% of them, there is evidence of weak splicing at this about 1% psi in other samples. So that's the idea. I'm gonna skip that. Okay, now the third and last criteria is monoallelic expression. So the idea here is to reprioritize variants that are deleterious, heterozygous, but actually turned out to be monoallelically expressed. We are using a test which we developed already back in 2017 based on negative binomial. And you may have heard about another test developed by Mohammadi Peshman and called ANEVA DOT. So the idea that we implement is the following. We count our reads for the alternative allele, as well as the read on the transcriptome for the reference allele. And then we test using a negative binomial test to count for overdispersion, whether there's a significant difference against a null hypothesis where the two alleles would be at the same amount. We also combine these together with an effect size. So we require at least 80% of the reads to come from this alternative allele. And of course, we prioritize the rare variants. So this is helpful and gives us complementary evidence. And here is an example where we can use it for diagnostic. So this gene, NFU1, was found to be monoallelically expressed in an individual. It is a well-known mitochondrial disease gene. From the whole exome sequencing, we had found the variant, but it was heterozygous, missense, probably damaging, so it was not prioritized. When you look at the RNA, you find monoallelic expression of that variant. You also find it, as you can see here, this is an outrider, so an expression outlier data. It is reduced, again, by something near 50%. So that suggests that the other allele is not expressed. And when we revisit the genomes now with whole genome sequencing, we realize that the other allele has a deletion on 1 exon. So now we have 2 variants in a compound heterozygous fashion. And that allowed us to then start further data not shown here based on proteomics, we could functionally nail this case. So altogether, as I said, we have diagnostic rates now of about 15% with these combining all those strategies. You may appreciate that you get actually diagnostics, not always by just a single technique, but sometimes by the combination of the two. For instance, there's a large overlap of aberrant expression with aberrant splicing and aberrant expression with monoallelic expression, giving you some further evidence that something is bad is happening on one of those genes. I'll finish with this talk with some practical considerations when implementing RNA-seq-based diagnostics, which will then relate indeed to this protocol paper that we just got accepted in Nature Protocols. The first point that may have somewhat prevented the field to embark right away into RNA-seq or massively into transcriptome is the worry that most of the disease we are looking at are showing symptoms in particular tissues, which may not be the accessible tissue. It may not be your blood samples or your skin fibroblast. I'd like to remind that symptoms are not causes, and that what is the affected cell type or the affected tissue doesn't have to be the, if I may use this word, affecting cell type, right? There are, for example, in coronary artery disease, there are metabolic defects that can lead to a heart attack, but are not caused by mutation, by a molecular defect in the heart. And this is true for many diseases where there is organ crosstalks, and a problem in an organ can lead to actually a defect in another one. It can also be the case for rare disorders. Another thing that you want to keep in mind is that maybe let's use this diagram here. You have to think, why do you have tissue specificity over variant? Variant is in DNA, it's in every cell. Why do I see an effect, particularly in the tissue? Well, it's because from the DNA all the way to your function, you have multiple steps, and somewhere tissue specificity kicks in. One hypothesis, and you have epigenetic silencing of a locus. A gene is not expressed in other tissues, it's only expressed, let's say, in brain. And then, of course, the variant will not really affect its effect elsewhere. So if this is the case, if we are not seeing in our sample tissue the gene expressed, indeed, we can't do much with RNA-seq-based diagnostics. But the effect can come later on. It can come at translation. It can come when the proteins find other proteins in specific cell types. It can come later on, even that a particular complex has a specific role in a specific cell type. It's very true for mitochondrial disorders, where mitochondria are particularly severely affecting tissues that are highly demanding in energy, but it's probably true for many other diseases. So you will not get all the genes on a clinically accessible tissues, but maybe you'll have enough of those expressed to actually improve your diagnostic rate substantially. And this is a question that many groups have been investigating. I want to cite here the paper by Gonoraski et al., who provides a web page where you can select a tissue and look at your sets of genes and find where they are expressed. Also, Barash labs in this paper here have done such an analysis for splicing. This plot here is from us. We are looking at different classes of genetic disorders, as well as all expressed genes. And we are looking at multiple clinically accessible tissues, as well as combination of those. And what you can appreciate is that for most of these rare disorders, we have already in blood the majority of the genes expressed. And fibroblast seems to be a pretty decent tissue to work with. So maybe don't be shy and do RNA-seq with what you have at hand. A clinical workflow would look like this. You start from your individual with a rare disease, prepare your samples. RNA-seq is done pretty quickly nowadays. And we've put together a workflow that integrates all these steps that I mentioned to you. That runs in about 2 days for a cohort, like 100 samples or so. This workflow drop include also some quality control steps, such as making sure that your genomes and your transcriptomes are matching by variants. It can happen that you have sample swaps, checks for sequencing depth, and then also, of course, execute all those modules. And then we have a web interface where you can combine different sources of information to help you prioritize your cases and your genes. Sorry. So this is a zoom now in the bioinformatics workflow. It starts directly from the raw file. You have basically a YML file where you define a bit all your data sets. You start from the biome files from RNA-seq, the VCF files from DNA-seq, and then run all of those modules. Can run on a parallel backend, and then we get all our outputs that we can browse and use to analyze your cases. There are some minimal requirements for sample size and sequencing depth. So we've done a few sample size studies in Outrider and in Fraser. We found that you would need about 60 samples to start to have enough statistical significance to detect aberrant expression. For splicing, you can go a bit lower at 30 samples on. One thing that we realize is that we tend to sequence a bit deep at 85 million reads per sample. We may be able to go a bit lower, but if, as we've seen by some colleagues, go down to 30 million reads per sample, then you lose quite a bit. Almost like 50% of your splicing outliers are then lost. So you need to go deeper. To help the community to start, and thanks to colleagues already using DROP, we put together a way to share count matrices. So if you don't have already 60 samples, but you want to embark on this, you can use pre-computed read count matrices that do not reveal genetic information, but allow you to start. And so we are starting this collection. Yeah, feel free, or please help us extending it. And we do this by tissue, as you see, and also by strand specificity of your protocol. We're providing resources on what I showed you for data, also on software. So this entire DROP pipeline is available on our GitHub repository. We're also running a workshop at the ASHG meeting since 19. And so the material for the workshop is available here. You have a notebook that allows you to test the code and see how this works in practice. So to conclude, I hope I could convince you of the added value of RNA sequencing over exome sequencing and genome sequencing. My talk was not a review of the field, but there are many other groups that are working on this project. We were developing this at the same time that Daniel MacArthur lab. So you may know this very nice paper from Barry Cummings, also from 2017. And other groups have continued on this idea. Steve Montgomery looked at using blood, so non-affected tissue for problem-affected tissues, and also saw that you can still get some decent amount of diagnostics. Gonowaski et al. Has looked at performing transdifferentiated cell lines and increased this way as diagnostic rates. And then Murdoch and colleagues have used DROP and showed how they could accelerate the diagnostic rates on their site. I hope I could convince you that these weak splice sites are some kinds of Achilles heels of the genome, places where The human genome is sensitive to variants and can lead to actually loss of functions on genes. And so those sites are outside the classical annotations, and in our opinion should be looked at more seriously. We are embarking in different projects now together with Prokish on proteomics-based diagnostics that seems to be a bit complementary to, quite complementary to transcriptome. Still in the process of wrapping a study. On the computational side, we are developing single-cell transcriptome, and we're going to be looking for colleagues that actually perform single-cell transcriptomes that might be important for some disorders, for example, in blood, looking at immune cells. So we are developing the techniques. We would like to have actual relevant rare disease data sets for that. And then, of course, there are applications of what we do in oncology. There's a lot of aberrant expression studied in oncology. So here, I think crosstalk between the two communities will be helpful, and to see if we can improve further our tools. And then I'd like to finish with maybe a call to the community. As you saw, we like co-developing, sharing data. And so if you want to join us, maybe reach out, and we could do maybe some regular seminars around RNA-seq-based diagnostics. Along the same lines, we will be in the next whole exome sequencing, whole genome sequencing subgroup of the HEI COVID-19 on February 4th, giving a short introduction to our technologies, trying to apply them on transcriptomes from severe COVID-19 patients, looking for potential rare genetic predispositions that would help us to interpret loci that would come up also with burden tests or so. So if you guys are collecting samples of severe COVID-19 cases, especially cases that would not, from young individuals with no comorbidities, I think here would be nice to collaborate and share data and do a joint analysis. With this, I would like to thank you for your attention, and the people who worked on what I showed today. Felix Breichmann developed Outrider. The first study was with Laura Kremer by Holger Prokisch, as well as Daniel Bader and Christian Mertes. Daniela Andrade and Michaela Müller have been actively working on DROP together with Vicente Yepes, who also wrote the entire protocol paper. Ines Scheller and Chris have worked on Fraser. And I would like to stress again, of course, our very nice collaboration with Holger Prokisch and our funding agencies. Thank you.
All right, thank you so much, Julien. Very interesting talk. We have one question from the audience. Boschen, can you ask the question yourself?
Yeah, sure, no problem. Nice talk, Professor. This is Boschen Zhao from Boston Children's. So my first question is that for the deep intronate mutation in the TIMMDC1 gene, did splice AI or MM splice predict that mutation trigger a new exon or pseudo exon?
So that's yes, MM splice not, because it's outside an exon, and I don't recollect the result for splice AI, but I think not, because it's not a splice site creation.
Yeah,
it is. But we should check, right? It may predict the splice site's improvement of the splice site before.
Okay, and
I don't have the answer now.
Yeah, actually, a follow-up question is that actually I like the idea of how to prioritize the mutation that located in the deep intron. Your idea is to, if the mutation overlap with the region that have naturally occurred pseudo exon in the human population, then a single SNP may further strengthen the splicing signal and trigger a pseudo exon. But I noticed that those naturally occurred pseudo exon usually occur at a very low level, so it's really hard to detect. I'm wondering whether there's any existing annotation or database for those region to facilitate us to do this kind of analysis.
We are doing this. Indeed, it's not trivial. There are data sets available. There are very nice resources. We have redone it using GTEx.
Yeah.
And we're in the process of wrapping this study. Indeed, it does work, but there's a lot of tedious work to get to the quality you need to apply this concept. But we've been talking about this since 2017, and it's only now that we're getting the results.
I guess so. It requires a pipeline of high sensitivity and specificity.
Yeah, there's a lot of cleanup to do.
Okay.
But then you can do that when you can do this aberrant expression. If you're interested, you can reach me out by email afterward, and we could inform you whenever we have the preprint also. Or just stay tuned.
Yeah, thank you very much.
You're welcome.
All right, we have a lot of questions from Ben Wisberg. So Ben, can you ask a question?
Yes. Hi, Julien. Thank you for the talk. I just wanted to ask if you could describe the step in the workflow. I don't believe you mentioned it in this talk, but where you inject outliers to determine the dimension of the autoencoder. And how does that work, and sort of why is that necessary? And I don't know if maybe this would be too technically sort of low level, but I'm kind of wondering about that as a comparison to sort of the peer factors, and how the number of peer factors is optimized in, for example, EQTL studies, or if that's sort of...
Okay, I'll try to say nothing bad, nothing wrong. How it works in our sites, I'll do it for Outrider. You substitute the read count you have with a read count that is away from the observed one by some standard deviation in the log scale, right? So you add a pseudo count, you go to the log scale, and then you go away from the observation by some amplitude that is a certain amount of your standard deviation. Then you run something that in principle is similar to peer. It's not exactly peer because we have a negative binomial loss and not a least square loss. And the way we're gonna learn the dimension of this latent space is by optimizing the precision recall curve of calling those points as outliers. Peer instead is using, so it's not the AIC, it's using a statistical criterion, which is called, I forget it now. It's not the AIC information criterion, but it's something based on not basically based on a kind of empirical cross-validation as this outlier detection method is, but it's based on the goodness of fit. And so that might be too confident in the modeling assumptions. That's one potential problem. And another potential problem with peer is there is this kind of sparse prior, and that is not super robust. So what we saw, and I hear that others seen the same, is that the number of latent component you get by peer is quite unstable from one run to the next, or by changing slightly the data set. We have something that is a bit more stable, but turns out to be a bit more stable. I hope it answers your question.
Yes, thank you.
Okay, I have one or two questions. So the first question is that for the job pipeline, can we actually accept data from different sequence platforms, like say less seq, high seq, lower seq. So that matters, to do the analysis.
If you're based on short read sequencing, you should be relatively comparable. Indeed, you're better off with all your data from your one platform. But what we saw is that many labs want to start, but don't have 60 samples to start with.
Yeah.
And so we suggest to try, and our idea is essentially, in the worst case, if your samples, let's say first 10 samples are really not comparable with those, the count matrices we are sharing, then you will have so many outliers that you will just not follow them up anyway. But if you're lucky, you already have some good evidence to start with. Of course, you don't write your diagnostic letter just from transcriptome analysis. You back it with your genome, your variant interpretation, and with functional evidence. But we believe you can start with this. And we have actually done simulations using samples from Crema and GTEx as control. And that allowed us to retrieve some of them. Vicente, do you have some you want to comment on
yeah,
that?
so in both cases, we record more than 70% of the confirmed either splicing or expression outliers that we knew that was also corroborated with either loss of function variant or splice region variant. We combined from these, the samples that we prepared in Munich, sequenced in Munich, with the ones from GTEx, and we were still, as I say, able to retrieve more than 70% of both expression and splicing outliers. So
you have at the cost of getting more outliers, right? You get more outliers when
yes.
you do, but it's still manageable to go through your hit list.
Mm.
The details are
one request. Yeah, sorry. So one requirement to run this pipeline is that, except for the sample size, and also you mentioned about the sequence depths, just want to double check. So, if I remember that correctly, you mentioned that the minimal sequence depth should be 85 million reads per sample. Is that right? I mean, if that's correct, then the sequence depth is quite high. I think 85 million reads per sample. That should be, the coverage I think is pretty high. For my understanding,
It's correct. It is higher than usually done. But when you come to splicing, this is what you have to see. If you look at expression outliers, you're using all the reads of a gene. So you're quickly into the high counts range. But if you're into splicing, you are just at the reads overlapping that splice site. And that's the criteria. It's like, what coverage do you get from your splice site? And so we have not explored all the sequencing depth. Maybe we should do this a bit more. 85 million is probably too high. Now in the latest grant, we asked for 60 million, and we think that's going to be fine. But the 30 million was definitely too poor, and we lost half of the calls by going down to 30 million.
Okay, see. Thank you.
The key point is this coverage
so
of
Chris,
yeah, go ahead.
Oh, sorry. Go ahead. I think Chris, do you have any question from your side?
No, I just want. I thought. First of all, it was a great talk, and I appreciate it. And I think I appreciated the fact that you said you pointed out the caveats to RNA-seq data in clinical use, but also the fact that we're not going to be perfect in any technology that we use, and so. You point out the caveats, but that it does have particular areas where it can be really beneficial. So I hope that becomes more readily used. So thank you.
Thank you.
Thank you so much. I don't think we have any more questions. Thank you, Julien. Really nice talk.
Thank you.
Thank you, Chris.
Thanks a lot.
All right.
Then bye.
Bye.